Good morning, dear students. Today, our topic of discussion is medium requirements for the fermentation process. In this presentation, we are going to see how the microorganisms are going to utilize the given medium for their better growth. Generally, the most useful and relatively simple primary classification of nutritional categories is one that takes into account two parameters. One is the nature of the energy source, and other is the nature of the principal carbon source. So these two disregards requirements for specific growth factors. Whereas phototrophs use light as an energy source and chemotrophs use chemical energy as their source. All microorganisms require source of energy, carbon, nitrogen, mineral elements, and possibly vitamins, growth factors if they are aerobic. Generally, the composition of biomass, if you see, they vary from molecules to elements, whereas the molecules like protein 30 to 60% weightage, carbohydrates 5 to 30% weight, lipids 5 to 10% by weight, DNA 1%, and RNA 5 to 15%, whereas ash includes the phosphorus, potassium, magnesium ions, etc. On an elemental balance, the carbon requires 40 to 50%, hydrogen requires 7 to 10%, oxygen requires 20 to 30%, nitrogen 5 to 10%, phosphorus 1 to 3%, and whereas ash generally 3 to 10%. If you see typically a composition of a biomass, that is C H 1.8 0.5 N 0.7. Suppose 1 kg of dry biomass contain 5%, what could be the amount of organic matter in a carbon mole biomass? This can be solved in a systematic way with stoichiometry of cell growth and product formation. If you see the different kind of organisms like autotrophs, where they use carbon dioxide as the principal carbon source, and heterotrophs, they use organic compounds as the principal carbon source. Whereas the photoautotrophs, which use light as an energy source, and they employ carbon dioxide as the principal carbon source. These include higher plants, eukaryotic algae, blue-green algae, and certain photosynthetic bacteria like purple-green sulfur bacteria. Whereas photoheterotrophic organisms, they use light as an energy source and employ organic compounds as a principal carbon source. These include photosynthetic bacteria, few eukaryotic algae. Whereas the chemoautotrophs use chemical energy sources and employ carbon dioxide as their principal carbon sources. Chemoheterotrophs use carbon energy sources and employ organic compounds as principal carbon sources. For example, fungi and most of the bacteria. Whereas the respiratory organisms, which couple the oxidation of organic substrates with the reduction of inorganic oxidizing carbon acceptor, usually oxygen. Whereas the fermentative organism, which energy eats from the organic substrates which are not so coupled. So generally the requirements of a nutrient media by the organisms, so requires a fruitful production of maximum yield of product or biomass per gram of the substrate utilized, and it will produce maximum concentration of product or biomass. It will use maximum rate of product for formation, and there will be a minimum yield of undesired products, and it gives the consistent quality and readily available throughout the year, and it also causes the minimal problems during the media making and sterilization, and causes the minimal problems on the production processes, particularly in the aeration, agitation, extraction, and purification and post-treatment processes. So usually the fermentation media, which is defined as, it is generally a liquid media referred as the broth, although some solid substrates fermentation are operated. The constraints of the medium that includes the water, carbon source, nitrogen source, phosphorus and sulfur source, minerals and trace elements, vitamins like biotin, riboflavin, oxygen, buffers, antifumigants, precursors, inducers, inhibitor compounds. So all this is a good recipe of medium. So that is being used by most of the organisms. If you see the table that explains the physiological functions of principal elements. So on an elemental level with their atomic number and what is their physiological function in the cell. Say for example, if you see the hydrogen, so which carries the atomic number, atomic weight of 1, which is the basic constituent of all the cellular, water and organic cell materials. Whereas a carbon, which is having atomic weight of 6, which is a constituent of organic cell materials. So likewise, if you see all the different elements present in the medium have a very good important functions in their cell physiology. So range from phosphorus, sulfur, chlorine, potassium, calcium, magnesium, iron, cobalt, copper, zinc, molybdenum. So this is a thorough and very good recipe for all the organisms. If you see typically a nutritional requirement for an organism, which is based on the elemental requirements, which satisfies a typical stoichiometric equation where a carbon source plus nitrogen source with an oxygen and sufficient minerals and other specific nutrients would give rise the cell biomass product, that is plant, carbon dioxide, water and the heat. So apart from this, there is a specific nutrient requirements that are there for auxotrophs, which they use a complex medium for their growth. So these are some of the common examples of media. So towards the production of very good important fermentative products like itaconic acid. So which uses cane molasses as a principal carbon source, which is a sugar, and zinc sulfide in the different hydrate forms. Whereas towards amylase production, so it uses these ground bean soya meal and brewer's yeast fractions, distillers, dried solubles, and lactose, magnesium sulfate heptahydrate, and so on. This makes a good recipe for the amylase enzyme production. Whereas the other antibiotics like ivermectin, so they use the cellulose as the important carbon substrate, and peptinized milk, autolyzed yeast, polyglycol, distilled water, and there should be a requirement of a pH of 7. You see on the right side, each and every component involved in a particular fermentation media with given with grams per decimeter cube. And some of the fermentations, they talk about the percentage of the components present in the fermentation medium. And similarly for endotoxin, this is going to be a endotoxin from Bacillus thuringiensis. They use the molasses, soya flour, different hydrogen phosphates and magnesium sulfate, and then ferrous sulfate, calcium chloride. All these are the important thing, and along with anti-foam agents. So this is a good perfect recipe for the endotoxin production from Bacillus thuringiensis. So apart from this, there are clavulanic acids production, the glycerol, soybean flour, potassium hydrogen phosphate, and starch, and many other requirements that you can clearly see here in this list. Similarly, for one of the important organic acid called gibberellic acid, so for this also there is a list of different carbon, nitrogen, and other trace elements present. And important amino acids like glutamic acids, for this also there is a requirement of a good amount of the carbon, nitrogen substrates. And it requires a growth factor called biotin, and also penicillin is used as an antibiotic towards glutamic acid production. And whereas the penicillin production, which depends on the glucose or molasses, corn steep liquor, phenylacetic acid, and lard oils, so which is the complete recipe for the production of antibiotic as per the different literature reports. And whereas if you see the table which gives the elemental composition of the bacteria, yeast, fungi on dry weight percentage. So each of these organisms, say bacteria, yeast, and fungi, so based on their elemental carbon, nitrogen, hydrogen, phosphorus, sulfur, potassium, calcium, magnesium, chloride, iron. So they vary a principal range of carbon sources. And if you see, typically the bacteria has high amounts of the carbon and nitrogen rather than the yeast and the fungi. Similarly, the other table shows how much of the yield coefficient that you can obtain from